Good morning, everyone. I must apologize for this delay. It's entirely my doing. Actually, I was only testing your patience, and you have been very patient. So, thank you. Thank you all for waiting this long. In fact, I thought it'll just be the chairs and Arka here and Kumar, and I'll speak to them only. But thank you all for waiting so long. So, as Kumar said, I got interested in ophthalmic genetics and molecular genetics way back in 1980. And at that time, some of the recombinant DNA techniques and molecular biology was just coming on stream, relatively new. DNA sequencing had just been done. Frederick Sanger, he sequenced a piece of DNA, and that was 1977. Ed Southern did his Southern blot, which we used for characterizing parts of the genome to look at the structure of the genome. So Southern blotting you've probably never heard of because then PCR took over. So Ed Southern described his Southern blotting in 1976. So things were just starting to happen. You'll have to be a bit more patient, I'm afraid. Maybe I can tell you a joke. There's this Irishman who went for an interview with his uncle. And the nephew went in first, and he was asked one or two questions, and then he says, okay, if you didn't have eyes, what will happen? So the nephew said, oh, you'll go blind. So he said, yes, good answer. Thank you, the interview is over. And as he was walking out, he said quickly to his uncle, the answer is blind, because he didn't have enough time. He was being led out of the room. So his uncle then went in, and he was asked one or two questions. And then he was asked, if you don't have ears, what will happen? You're blind. If you don't have ears, you'll go blind? How is that? So he said, yes, think about it. Because if you don't have ears, your hat will drop in front of your eyes and you can't see anyway. Because your ears keep your hat up. Because you know the bowler hat, which is kept up by your ears. So he said, if you don't have ears, then naturally your hat will. So anyway, what I wanted to say is that you should always think about sight and sensory perceptions as a polygenic condition, because it's not the primary thing which relates to the phenotype. Even the ears are important for the eyes. So anyway, I think I'm back on stream, so I can stop telling my silly jokes. I have a few more. If you're interested, I'll tell you a few more afterwards. Okay, so as Kumar said, I was mainly at UCL. But for the last 7-8 years, I've also been director of the Andalusian Center, which is in southern Spain, in Seville. And it's a center for molecular biology and regenerative medicine. Place a call. Who shall I call? Yes, do you see that program Amitabh Bachchan does, I think? And you can call your uncle or whoever. I can run it from here, no? Yeah. Right, okay, yes. So let's begin. So, as Kumar said, I've mainly worked with ophthalmic genetics. And when I started, one of the major vision problem as a monogenic condition is retinitis pigmentosa. And I don't know how many people know the background to retinitis pigmentosa. So I thought I'll tell you a bit about the background and what is the clinical problem. To put it in context, then the genetic work. And what happens with retinitis pigmentosa is it possible to remove this or shift it to one side? So with retinitis pigmentosa, the patients at some point in time, not from birth, but could be in their teens or later, they start to lose vision, initially peripheral vision, and then later on central vision as well. And this happens, is there a pointer? Remember, the first on the left-hand side was the normal retina, and yes, so that's a normal retina. And a patient with RP, they start with problems around the periphery, as shown here. You see this black pigmentation around the periphery. The central retina is still okay, and if you were to do an electroretinogram, this is the normal, and then somebody with RP will start to lose that activity, and eventually when a lot of the retina is affected, the ERG becomes quite flat. So the initial symptoms starts with night blindness because the photoreceptor cells maybe I can do without the loudspeaker. So the photoreceptor cells in the retina, there are two types, rod cells and cone cells. So rod cells allow you to see in dim light. And patients with RP, they have problems because it's the rod cells that malfunction and eventually die. And because of that, you get peripheral loss of vision because the rod cells are mainly concentrated around the retina, and cone cells are here mainly in the middle. So they have quite okay vision till quite late in life, as long as the middle bit is okay. And then eventually, when a lot of rod cells are dying, then even the middle is affected, and they eventually go completely blind. So these are the symptoms of RP. Of course, normal vision, you can see everything, but somebody with RP is also known as tunnel vision. So central vision is okay, but peripheral vision is gone. Now, how is that happening? So here is a normal eye, and the retina is the very back layer in the eye, and here is a histology picture of the retina. And I've shown as a schematic the different cell types in the retina, starting with this is the direction of light, as you can see. So the very first cells which come in contact with light are the ganglion cell layer. So in this slide, it's like this. So light is coming from the top. So on the top are ganglion cell layers. Then in the middle are the bipolar cell layer. And then finally, the third type of cells, which are the photoreceptors. And photoreceptors are two types, the rod-like cells here, and then conical cells. So red, green, and blue, these are the primary colors. And you can see the color vision is facilitated by these cone cells, so rods and cones. And in RP, mainly the rod cells die. Now, in the human retina, there are something like 100 to 110 million rod cells, and between 6 to 7 million cone cells. So the cone cells are outnumbered by rods by 20 to 1. So in RP, it's the rod cells which die in such large numbers. So eventually the cones are also killed. So in the retinal tissue, ganglion cell layer, bipolar cell layer, and then the photoreceptor nuclei are here. The light blue area is the inner segment of a photoreceptor cell, and the dark blue is the outer segment, and then below that is retinal pigment epithelium, which I will show you in a better slide here. Oh, this one, can you see? I can see very clearly on my picture, but it's very dim here. Do you know, is it some way to brighten it up? Or you can see the cartoon diagram with the same for the other slide also. No, the other one's okay. Yeah, these are okay. So it's just, sorry about that. But basically, it was in a more detail what I'm showing in the cartoon diagram, which I think you can see. So the ganglion cell layer is here, bipolar cell layer is here, and then the photoreceptors. And there are two types of photoreceptors, the rod-like and cone-like. And the tips of the outer segments of rods and cones, they interdigitate with RPE, retinal pigment epithelium, which is down here. And believe it or not, almost 10% of your outer segment from every photoreceptor cell, they're shed and ingested by RPE and degraded. So therefore, the outer segments of both rods and cones, they are renewed on average every 10 days, because you lose about 10% every day. So in 10 days, the entire outer segment is renewed. And this is an electron micrograph of a human retina. And you can see the fat cells, these are the cones, conical, and surrounded by, as I said, 20 to 1. They outnumber cone cells by 20 to 1. So these are the rods. A top view of the retina, these are the cone cells, and you can see how the rod cells completely surround them, 20 to 1. Then here they are, all standing up in military fashion. These are the rods, and in between, now and again, you'll see a cone. So here is a cone cell, you can see. Here is a cone cell, you can see. So like that. Now, lying down, and you can see this is a rod cell. So this is the outer segment, this is the inner segment, and between the outer and inner segment is the connecting cilia over here. So every day, a lot of protein is transported to the outer segment to create the discs in the outer segments. And I'll talk about the discs a little later. So the outer segment is composed of mainly discs. So imagine a packet of biscuits, and each biscuit is a disc. And it is in the disc membrane that you have the photopigment. So in case of rods, it's rhodopsin, and in case of cones, it's conopsin, so red, blue, and green. So that's here. And the initiation of phototransduction, so this is the phototransduction cascade. It's quite a busy slide, but the purpose of showing you that is here is rhodopsin, which I just mentioned, which is in the disc membrane. And as a photon of light hits rhodopsin, it initiates the cascade of you being able to see. And a photon of light will make rhodopsin photoexcited, which in turn will interact with transducin. Here is transducin. And transducin will get activated, and transducin will then interact with phosphodiesterase. And the beta subunit of phosphodiesterase will then convert GTP to cyclic GMP. And let me start again. So cyclic GMP is bound to the channel protein and keeps the channel open here. So sodium and calcium can come into the outer segment. And as the photon of light activates rhodopsin, transducin, and phosphodiesterase, then phosphodiesterase will in turn convert cyclic GMP to GMP. And as a result of that, the channel will close. That initiates the process of vision in each cell, in each photoreceptor cell, rods or cones. the same thing is happening. And the electrical signal generated here will go back to the bipolar cells, and from bipolar cells to ganglion cells, and then the nerve endings of ganglion cells will come together and form the optic nerve, and will take that signal back to the visual cortex in the brain. Okay? So the entire process of phototransduction, starting with the photon of light, this is what happens, and this is happening in every photoreceptor cell as it sees a photon of light. Okay? So that's the biochemistry. So I've shown you the structure of the retina, the principal cell types in the retina, and for our purpose, in terms of retinitis pigmentosa, it is the photoreceptor cells, the rods and the cones, and then the biochemistry of photoreceptor cells, how it functions and how it's converting the photon of light into an image in your visual cortex, and you're able to see the world. In dark light, in dim light, in starlight, or when you go out for a walk when the full moon is on, it's mainly your rod cells which are working. But in bright light, in daylight, it's the cone cells which are working, and therefore you can see the color vision and you see the world in color. Okay, so that is what I was trying to tell you by the background and the problem in retinitis pigmentosa, because in patients they're losing the rod cells, that's why you're getting tunnel vision, and finally even the cone cells die, and blindness. Now, before the advent of molecular techniques and DNA techniques, the patients, when they came to the clinic, unfortunately the ophthalmologists couldn't really do anything. All they saw was that the retina, there's black pigmentation, and gradually the retina is degenerating and people are going blind with RP. And it's quite a burden for the society because it affects mainly the working age group people who are productive for the society. And if they have RP, it's a devastating condition for them, and blindness. So with the advent of recombinant DNA techniques, for the first time, we had this opportunity to use reverse genetics. And we could go back and study the family and track the gene. So this is what was possible. So I'm jumping the gun here and showing you my result very quickly, first of all. So what you see here is a DNA band. So what was discovered in the early 80s was that if you took DNA, it was possible to detect restriction fragment length polymorphisms. So individuals, everybody in this room, were polymorphic for fragments of DNA. And that means that a genetic variation. And you could then tag that and you could use it for genetic studies. So you have a tool here of DNA markers, which are genetic markers, which you can then use in families and track the phenotype. So it could be anything, any disease phenotype, and in my case, it was retinitis pigmentosa. So here is the family with retinitis pigmentosa. What you see here, the mother was a carrier of RP, the mother, and she has 3 sons, 1, 2, and 3. 2 of them are normal, these 2 are normal. This one is affected. And 2 daughters, and they are both carriers of RP. They are carrying the RP gene, okay? So this little family has RP, it's got X-linked RP, it's clear from the inheritance pattern that it's X-linked. So the female has 2 X chromosomes. 1 X chromosome carries the normal RP gene, the normal gene. The other X chromosomes carries the RP gene on it. So unfortunately, she has passed on the X chromosome to this son with the RP gene on it, and he is blind. Fortunately, the other 2 sons got the normal X chromosome, and they are normal. But unfortunately, she passed on again the affected X chromosome to her 2 daughters, because the 2 daughters got 1 X from the mother and 1 X from the father. So both of them inherited the affected X chromosome, and they're both carriers. Now look at the DNA picture here. So the father has the upper band, on his X chromosome. The mother is heterozygous. This is the polymorphism. So this band has nothing to do with RP. This is not the RP gene. This is simply a marker which is very close to the RP gene. So the mother has two bands, the upper band and the lower band. And now let's look at the two sons who are both normal. So this one and this one. These are from these two normal boys. And both of them have inherited the upper band from the mother. So you can see the upper band here and here. So they're both upper bands and they are both normal. The RP gene is on the lower band and she has passed on to the affected boy the lower band, as you can see, and he's affected. Also, the two daughters which are here, they're both heterozygous, they both have two bands. The upper band comes from father. They've inherited the father's X chromosome, so the upper band is from the father in both of them. But unfortunately, they've inherited the lower band from the mother, and they are carriers of RP. So here, what I'm showing you is a very quick reconciliation of a DNA technique, DNA technology, because the genetic marker is a piece of DNA from the human X chromosome, able to detect this polymorphism. So from one X chromosome, it detects a higher band than the other X chromosome. And this genetic marker is perfectly linked with the phenotype, with the disease. So here in the early 80s was the tool where we could study families and link them to the disease. Now, obviously the interest was how to generate these markers. And that's where I first started in 1981, because this came later. But we knew the problem, we knew the background, and we had to generate the tools. We had to actually create the markers first. So we started by first isolating human X chromosome. So this is just telling you that a reverse genetic approach for the identification of genes, and you have to do disease gene mapping. And for that, you need to make chromosome-specific genetic markers. You do genetic linkage in pedigrees, as I showed you previously, and then you can clone the disease gene. So our target in 1981 was to create genetic markers for the X chromosome, because the families that we were studying were X-linked retinitis pigmentosa families. Now, we started with first isolating chromosomes, making a metaphase chromosome soup, and isolating the human X chromosome from it using a FACS sorter, but it was specially tuned so that it could sort chromosomes rather than cells. So it's a cell sorter, but we converted it for chromosome sorting. And we were able to, unfortunately, again, you can't see the bottom here, but these peaks show you the different aliquots from the FACS sorting with different human chromosomes in it. And this was the aliquot which had human chromosome 7 and human chromosome X was sorted. So we sorted something like 2 million chromosomes, 7 and X, made the DNA from it. You can't see anything. You just have to rely on faith and someone upstairs who says, okay, it's there, don't worry, the DNA is there. So literally from 2 million X chromosomes, you'd get something like 2 or 3 nanogram of DNA, that's all. So you had to be extremely careful how you handled it. We then digested that DNA. So here is the DNA from the chromosome, this separated chromosome, digested into small fragments. At that time, the vector I was using to clone these pieces of DNA, these were EcoR1 fragments. And this is lambda GT-Wes, the phage vector. And it had 2 EcoR1 sites. So when you digest lambda GT-Wes, you will get a 21 kilobase band and a 13 kb band. And by removing the middle fragment, you could clone your piece of human DNA in it. And therefore, you could create recombinant clones. So by taking human DNA from the chromosomes that we had isolated, creating millions of little EcoR1 fragments, we could clone the fragments into the phage and then package them into viable phage particles, these viruses. And that was a human X chromosome library. So in that library, we had millions and millions of pieces of human DNA attached to the virus, which we can then propagate and clone. And then our task was to isolate these pieces of DNA, these clones, which will hybridize back to the human X chromosome and act like a marker. So we did that again. I'm sorry you can't see the slide, but it's very clear here on the thing. But on these two slides, and here is the southern blotting, so maybe what you can see are, if you focus here, so here is the 21 kilobase band, here is the 13 kilobase band of the phage molecule, and then you will see in different clones, different human X chromosome fragments in them. And these are the different fragments that we were able to clone and isolate, and we could then use them as genetic markers to detect the polymorphism back on the X chromosome. And that's what we did. Yes, so here you can see it very clearly. So we had screened the X chromosome library, isolated these pieces of DNA, and now using one clone at a time, in other words, one piece of DNA from the X chromosome at a time, we could challenge it against a panel where the DNA was from a cell line containing 4 X chromosomes. This is DNA from a female with 2 X. This is DNA from a male. We know it only has 1 X. And this is a mouse-man hybrid, a somatic cell hybrid DNA, which has the human X chromosome in it. So it's against the mouse background, but retains only the human X chromosome. So here was the panel, and if you look at it, it's the other way around. Here is the DNA with the 4X, so in other words this, then the female with 2X, then the male with 1X, and here is the human-mouse hybrid. So these are 3 different examples of pieces of DNA coming from the human X chromosome, which we could now use as a genetic marker to track phenotypes coming from the human X chromosome in families. So these became the genetic markers which we used. And I showed you, so I'll skip a couple of slides here and come back to there. So this is using one such marker from the human X chromosome, which detects a polymorphism, and we could then study the segregation of the marker. Remember, the marker is from the human X chromosome, so we know that the disease gene must be close to this marker, because in going from one generation to the next, if the marker and the phenotype, if they stay together, that means the marker is very close to the disease gene. And therefore, you can do positional cloning to isolate the disease gene finally from the X chromosome. And that was the positional cloning of disease genes at that time, because we knew the problem. We had no idea what the biochemistry was. We didn't know what the protein was, which was defective. So the only way you could do is take a reverse genetics approach, map the gene, clone the gene, and once you've got the disease gene in your hands, you could then ask the proper questions. What is the protein? What is going wrong? Why it is causing RP, etc., etc., etc. So that was our strategy way back in 1981 when we started the project. And we were extremely fortunate that, again, now here, to clinicians and genetic nurses and counselors who are present in the audience perhaps, that the importance and value of collecting large families, because when you have large families properly clinically characterized, so here is an X-linked RP family and lot of affected boys, as you can see here, in different, who are cousins, and using that same marker, which I showed you in the previous slide, showing linkage, using that same marker, you see now the affected boys have got the A2 allele. A2 allele means the lower band, okay? That's the A2. And A1 is the upper band. And all the affected boys have A2, and all the normal boys, like here, has A1. So this is the same marker, and in such a large family where during going from one generation to the next, if the marker was too far from the disease gene, then crossover will happen and they will separate. But it didn't. In all these meioses, which are independent meioses, transmitting the genetic marker and the phenotype, there was no crossover. So in other words, the marker was perfectly linked with the disease phenotype, and we were able to map X-linked RP, and we published it in 1984. And the X-linked RP gene, here was the marker L128, and the X-linked RP gene was very close, and eventually the X-linked RP gene was cloned. In fact, as it happened, as it turned out, there were two X-linked RP genes, one here and one over there. And as my host today, Dr. Kumar, who introduced me so kindly, he did his PhD with Michael Denton in Australia and New Zealand. And Mike Denton was in Australia at the time, and he mapped the second X-linked RP locus, which was called RP3. And we called ours RP2, because at that time there was one RP1, which was supposed to be autosomal dominant, and nobody knew where it was, but that was RP1. So we called ours RP2 after that, and that was the mapping of X-linked RP2. And soon after us, Michael Denton published mapping of RP3. So there was genetic heterogeneity even on the human X chromosome. So I've now described the process of linkage to you, and we are on the way where by mapping disease genes, and this then started happening across the board. So in the early days, mapping of Huntington's disease, polycystic kidney disease, and many other genetic traits, monogenic genetic traits, where bit by bit started being mapped. And Duchenne muscular dystrophy, which the disease gene, DMD gene, is here, and that was identified, mapped and identified and cloned in 1986 by Tony Monaco and Lou Kunkel at Harvard in Boston, and they found the DMD gene from there, but the same technology. So at this point, I'd like to tell you about, we'll move on and I'll go quite fast from now. So really the major work and a pioneering work was done by Peter Humphreys in Dublin. And in Ireland, they are Catholics and they believe in large families. And Peter was able to collect a lot of dominant RP families, large RP family. And in 1989, he published the mapping of dominant RP to chromosome 3 using this very large family, using a marker from chromosome 3. He was able to show linkage and was able to map it to human chromosome 3. It was a major breakthrough. You have no idea what it started at that point, 1989. And in our lab, we had by then collected quite a few dominant and X-linked pedigrees. And here are two examples. Again, I'm showing you these large pedigrees because what it did, if you have single pedigrees, that means you're tracking a single gene. You don't have to worry about genetic heterogeneity within the family. And if you can map it, you will map a locus, and you can then study the other families and see whether they map to the same locus or not. So we had these two dominant RP families, and phenotypically one was D-type with diffuse loss of function, and R-type, which is regional loss of function for the retina. And at that time, my clinical colleague was Alan Bird. He started the RP clinic way back in 1965 and perhaps collected the largest collection, more than 20,000 families in UK alone with RP, actually was seen through his clinic over a period of some 30 years. I started working, I was in Edinburgh in 1981, and he was my clinical collaborator for Moorfields Eye Hospital in London. And he was ably assisted by Marcel J. She is the one who used to track So when the patients came to his clinic, she used to then, after he has seen them, she would then start asking about the family tree. And she started building up those families. So these families, they took Marcel J 2 years, 3 years to build up. Because I had told her that unless the families are big, we will not be able to map them. And we can't pool families because one family could be from one chromosome, mapping to one chromosome, and the other family could be the other chromosome. For example, this one is from chromosome 3, and this one is from chromosome 7. So if you pool them, you'll never get linkage because it'll be just a hodgepodge. But within the single family, you could map it. So she started doing that, and here is the enthusiasm with which she would start. So here is her work. It took her almost 5 years to put this family together before it was studied. And you can see she even traced the will written by him, and this is John Norwood. He was born in 1728, and he wrote his will in 1785, and he signed it. So we think that he wasn't blind, that the RP came through his wife. But she had traced 9 generations, running in churches, and first of all, the birth and marriage certificate. There's a registry in London, St. Andrew's House, and there you can see, and you can go back to 1850s. But beyond that, you have to go to the church, the graveyards in the churches, and she would actually go and look at the headstones and try and connect it. And she did. She actually connected up this entire family. And these people in the subsequent generations right down here, who used to come to Alan's clinic, they had no idea about these people down there. How would they? Because they're separated by 9 generations, and they had no idea. And then Marcel and Alan will tell them, well, look, we have your cousins, distant relatives who are 9 generations separated, and a lot of them didn't want to meet them. They said, why should we meet them? But we could link them all up. And in fact, we were able to use that family for subsequent mapping. But coming back to the chromosome 3 story, this is what Peter did. Using a marker called C17, he was able to link it to this part of chromosome 3, and next to the chromosome was the gene for rhodopsin. So rhodopsin became the hot candidate. And here is the full rhodopsin, 347 amino acids long. It is a transmembrane protein. This part of the molecule is intradiscal. It is inside the disc. This is the transmembrane part, and this is the cytoplasmic part. It is this part which interacts with transducin. So initially we thought that we should see all the mutations in this part because that's the part which interacts with transducin. But later on, we discovered that mutations in any part of the molecule, cytoplasmic, transmembrane, or intradiscal, will result in dominant retinitis pigmentosa. Of course, at that time in the 1989-90s, PCR had just come along, and Mullis had described the technique, and everything was much faster than the old days, and we could amplify each exon of rhodopsin and sequence rhodopsin in individual patients, and we were able to trace. So first of all, rhodopsin itself, you can see the outer segment of photoreceptor cells, and rhodopsin is the major protein expressed in the discs, and you can see how they are stacked like a packet of biscuits, and the tip of the outer segment is then shed 10% and ingested by the RPE. So 90% of the protein in the outer segment is rhodopsin, 90%. And of course, if you have a mutation in rhodopsin, then that is bad news. And we were able to identify the very first mutation in Europe in that family, which was from chromosome 3, which was a rhodopsin mutation. And here is a little anecdote. Here is a normal sequence of rhodopsin, and here is the mutant, the mutation we discovered. It was a 3 base pair deletion, and that particular deletion happened here. So if you look at this part here, it goes CAT, CAT, and sorry, I should start here, CAT, 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 3 times. And my postdoc who discovered it, Chris Inglehorn, he was funded to work on retinal genetics, but not on human. he was funded to work on cat. There was a cat model of RP which was available in Cambridge, and we had that DNA from those cats, and he was trying to find the gene in that cat model, CAT, cat. And the very first human mutation he discovered spelled cat 3 times. So there had to be some divine intervention. So I always used to tease Chris that the first mutation you found spells cats, you better get working on the cats. So anyway, we found many different mutations in rhodopsin. So this is the rhodopsin molecule. All those red circles are mutant amino acids, and these are all the little families and large families where we had found the mutations. We could detect the mutations by PCR and just digestion quickly. And this is a very old list, shows about 50 different mutations, but the recent count of different rhodopsin mutations available, identified in different human families, it goes over 150. So out of 347 amino acids, 150 amino acids are mutated in dominant RP. Clearly, there's a story about evolution here, because in evolution, as each protein evolved and as it became perfect in function, nature doesn't want to keep repeating itself. It keeps it. So rhodopsin is seen from bacteriorhodopsin. Even bacteria, halobacteria, has rhodopsin in it. It's called bacteriorhodopsin. And it swims, the bacteria swims towards light, and it functions exactly like human rhodopsin. And that same sequence from bacteria to Drosophila to fly, and finally down to human through all the different species and so on in evolution, rhodopsin is very well conserved. And in fact, if you look at the human rhodopsin and cow rhodopsin, there's only 2 or 3 amino acids which are different. So nature knows its job, and once it knows the protein is functioning properly, it doesn't change it. So, of course, the role of rhodopsin is to do phototransduction, as I described to you before. And rhodopsin is the key molecule in the rod cells, and if you have mutations in rhodopsin, you would end up getting RP. Now, I'll tell you, first of all, let me ask you, how much time have I got? Because it's now already 11 o'clock. 15 minutes. Okay, fine. So I'll go triple fast now. So what I was going to show you were how you can link things once you had mutations in rhodopsin, and how we found that the transcription factor that switches on rhodopsin is called NRL, and mutations in NRL also cause dominant RP because it affects the expression of rhodopsin. And if you have a mutation in NRL, NRL doesn't function properly, and it gives rise to RP. So my next slides are to do with that, but in between I was going to show you how microsatellite markers and dinucleotide markers, how they became very useful genetic tools. And you can see here one such marker showing you don't even need to do lot scores. You have the family, and you see that all the affected individuals have that same band, and therefore this particular marker must be close to the RP gene. And that was the mapping of our large family to chromosome 7. So there's a dominant RP locus called RP9 on chromosome 7, which we mapped. Anyway, this was the story about NRL, and I'll skip it, except to show you that with the mutation, the NRL protein became more active. So it was expressing rhodopsin at a higher level. And if you already, it was known that if you express rhodopsin more or less, you will also get RP. So that fitted in very nicely, that a mutation in RL which made rhodopsin expression higher would also cause RP. Now, another thing which was fascinating for us is that you take it for granted that cells and tissue in the eye, retina, if there are problems with that, it will cause disease and retinitis pigmentosa. But what puzzled us and amazed us is that genes that are ubiquitously expressed all over the body specifically also causes RP. And how does that do it? So the next example that I was going to show you, and I'll again run through it very quickly because of lack of time, is this particular family, again a large family, and we mapped it to chromosome 19, and this work was done by Irenga Vithana, who is in Singapore now, and she was my PhD student, and as a PhD student, she mapped and physically actually cloned the region containing the Rp gene in it, and eventually discovered that this gene called PRPF31, which is a splicing factor. So splicing factors and spliciosome, the role of that in every cell is when you have a RNA molecule, you have to remove all the introns. And once all the introns are removed, it creates a mature messenger RNA, which contains all the exons. And this mature messenger RNA is then translated into a protein. So if you have a problem with splicing factors, and in the case of the retina and in photoreceptor cells, what if the splicing factor was not splicing rhodopsin properly? And this could be the answer. So we found mutations in the splicing factors causing only RP, retinitis, no other problem. So clearly, in every other cell in the body, it can tolerate mutation in one copy, but because the other copy is normal, splicing happens normally. But photoreceptors are uniquely tuned. They are the most active cell in the body, photoreceptors. And I think that the reason you get a retinal phenotype, an RP phenotype in humans, where the genes are ubiquitously expressed, is because the photoreceptors are ultra sensitive and they need everything completely normal. And if that is not the case, they cannot tolerate that 50% is mutant and 50% is active. Other cells in the body can tolerate that, and they don't have any other symptoms. But these patients, they have retinitis pigmentosa. And we were the first to discover these splicing factor mutation and ubiquitously expressed gene. So I'm going to skip all these slides because that is the story. And I'll come to this other gene, which also had nothing to do with splicing, but it was ubiquitously expressed. Here was a large family. We mapped it, cloned the interval, which was about 16 megabases in size, and there were 51 genes in that, 53 genes in that interval. We had to sequence more or less all of them, and finally we found mutation in this molecule, this protein, topos, in this gene called topos. It's topoisomerase binding protein. It is ubiquitously expressed, and yet mutations in topos cause RP. And how is that? So I gave you one example of a splicing factor which is ubiquitously expressed, and this one, what we discovered after we cloned the gene, that when we did immunolocalization, we found that it localizes in the connecting cilium between the inner segment and the outer segment. It localizes here. And yet, before that, all the work with topos, which the cancer people had done, it was all localized in the cell body, not in photoreceptor cells, but the cell lines they were using, it was in the nucleus. It's a nuclear protein, and it binds to p53 as well, etc., etc. But in our case, we found that in the photoreceptor cells in the retina, it localizes here. So we did a huge amount of work, more than 5 years of work, and eventually we were able to demonstrate that the reason it's localizing is in the connecting cilia is because it's actually acting as a motor protein in the connecting cilium between the inner segment and the outer segment. It's somewhere here, localized here, and is helping probably in the retrograde transport. So because I told you that every day proteins from the inner segment move to the outer segment, and then a lot of proteins which are degraded they move back and then degraded in the cell body. And it looks that topors is involved in the retrograde transport. And that's why, again, same protein, but having a different function in other somatic cells versus photoreceptor cells. So nowadays, we are not so surprised when we find that there are ubiquitously expressed genes causing retinitis pigmentosa alone. And it's because they have a different function within the photoreceptor cells. And finally, I like to tell you about the largest eye gene that we discovered. Again, through studying, and this is a recessive Rp gene, and it was the largest gene in the human genome, over 2 megabases, for which we had to actually sequence almost 6 megabases of the human genome from chromosome 6, because although the human genome had already been published, this work is from 2008. So although the human genome had been sequenced and published, but people who are working on the bench and very minutely in parts of the human genome, they discovered that many parts of the human genome had not actually been sequenced. So we had to actually sequence almost 6 megabases of DNA from this part, and eventually we identified the gene. And this is the position cloning, and this is the gene itself. And after we had cloned the gene, then through heavy-duty bioinformatics, we discovered that quite a while back in Charlie Zucker's lab, who works on Drosophila, they had identified the same protein called EYS. They named it EYS, eyes shut, because mutation in EYS causes degeneration in the Drosophila eye. And we discovered that our protein, when we characterized it all, our protein matched the eyes protein from Drosophila. Again, a very nice anecdote for evolution, because in evolution in the fly, such a large protein had been perfected for function, and in the Drosophila rhabdomere, it is involved in retinal architecture, because it's also known as spacemaker. Because what happens is that this is the Drosophila eye, and these are individual rhabdomeres, and the white stuff in between is EYS, or spacemaker, or SPAM, as we called it in short. And that is the same protein which is mutated in humans in recessive Rp, and that's the largest gene at the level of the gene. Is more than 2 megabases long. And if you delete EYS in Drosophila, then all the rhabdomeres, they fuse together, like shown here. This, as it happens, is the structure of the retina from bees. Bees do not express SPAM or spacemaker, they don't. So their rhabdomeres are all fused, and they have a single channel of communication with the visual cortex. In the Drosophila, there are 7 different channels. Each rhabdomere has its own channel of communication. And of course, you try catching a fly, it's impossible. A bee you can catch quite easily, but of course you have to be very careful about getting stung. But you see the difference here. And if you delete SPAM from flies, it'll look like that. If you express SPAM in bees, it'll look like this. So fascinating how evolution works and how you can play around even in live animals. So you can make the bee see like a fly. So then what we discovered when we started working on SPAM, that in mammals itself, SPAM has been disrupted. So it's present in the fly and present in humans, but when we looked at mouse to create a mouse model, it wasn't there. It was disrupted. In the mouse genome, SPAM doesn't exist. In the cows, SPAM doesn't exist. But it is present in the dogs, in chicken, in horses. And you see, if you look at a cow and a horse running together, the horse will spot something obstructing its path way ahead of the cow, and it'll go around it. Whereas the cow, running along, galloping along, and go poof, crash into the hurdle, because I think that's because it doesn't have spam and doesn't have the spacemaker. So I'm sure it doesn't have acute vision like the horse does. And anyway, so my story of RP genetics is over. Currently, more than 50 genes for RP has been identified just for retinitis pigmentosa. I think it's actually 60 genes spread throughout the human genome. These are different human chromosomes, and the RP genes are on different chromosomes. In terms of describing all of the different retinal genes, more than 200 genes for retinal dystrophies have been described, which you can download from this website. And here is a graph showing the start, and this is getting quite old now. At this point already more than 200 genes had been identified. I believe now it stands at more than 250. So I think I'll stop here, because I was going to, one final message. The most common single gene causing RP is obviously rhodopsin, all started by Peter Humphreys. And the biggest gene and a major gene for autosomal recessive RP is EYS, SPAM. And for X-linked RP, it is RP3, which was originally mapped by Mike Denton in Australia. And 358 different mutations. This is also more than a year ago. I think additional mutations in RPGR have been found. So this is for X-linked RP. This is the most common single gene causing X-linked RP. So I'll stop here, because I was going to tell you about gene therapy, but I'll stop unless you guys are madly interested in gene therapy. I'll just stop here and just leave it at genetics of RP. Thank you very much, and thank you for your patience. Hello, yes. Not that we can tell. RP patients, they look very similar. If you take dominant RP or X-linked RP or recessive RP, phenotypically they are very similar. And but what is surprising, because all these ubiquitous genes we have discovered with dominant RP, and we find they're ubiquitously expressed, but leads to dominant RP, same as rhodopsin or peripherin. No, nothing else, only RP. Yeah, and the phenotype is the same. Yes. And as I said, the only explanation is that the photoreceptors is so highly tuned that it cannot tolerate that 50% of the product is mutated. Yeah. Yes, because probably it's not splicing rhodopsin part of the time, and then you have got faulty rhodopsin. Exactly, because you are not getting the full dose of rhodopsin. If that is the mechanism, we still don't know what exactly the mechanism is for mutations in splicing factor. We know what the mechanism is for NRL, but NRL is specifically expressed in rod cells, so that's fine. But splicing factors are expressed everywhere. Yeah, and topos is expressed everywhere. So it must be something to do with the dosage, because it's only 50%. I thought you guys must be. Yes.
So amongst all these genes, do you see genes which are functionally haploinsufficient, where you have, say, heterogeneous mutation at the H level? So you have only one allele being expressed, so you have less
Yes.
protein causing a haplosufficient.
Yeah, I think, yes, I think sometimes, like in X-linked RP, the females, in some cases, they start showing the phenotype. And that could be A, due to the lionization, because you're getting 50% of the cells sort of lionized, or and 50% are because to maintain the same X chromosome dosage in males and females, one X is made redundant, lionized, and the other X is working. So if you take the overall picture in a female, if the mutant X chromosome is more in number than the normal X chromosome, then you will begin to get haploinsufficiency of the normal product. So that could explain that haploinsufficiency. But in a lot of dominant conditions, it is known that it is haploinsufficiency because one allele is mutant. At the same time, it's dominant negative effect because the mutant protein is attacking the normal protein and causing the disease. So all sorts of options are there, and each one has to be studied clearly. Yeah. Okay. So, right. Kumar is making you all sit through longer. So anyway, this is the next obvious question, that if you can identify, if you can do the genetics and you can find the genes and you understand the biochemistry, oh, I see. Let's see what happens. Yeah. Yes, that will be difficult. So, okay, so once you know your genes and you know the function of the protein, and you have an understanding of what is going wrong and what you can do, then clearly biological therapy is your one approach, where either gene therapy and more recently cell therapy, where you adjust, you genetically engineer the cell and you can transplant cells in the retina. And these biological therapies are probably the major route for treatment for patients with RP or other retinal degenerations. And when we had found these genes in the mid-90s, so I was very keen to pursue gene therapy as an approach and see whether we can right. So yeah, that's much better, isn't it? Yes. Right. So so the approach we wanted to explore in the mid-90s is whether gene therapy will work or not, because we know that rhodopsin is mutated, or we had discovered that mutation in peripherin, which is also expressed in rod cells, is mutated in some form of dominant RP. So there was this RDS mouse, retinal degeneration slow, RDS, and it was known that mutation in peripherin is causing RDS in the mice. So we thought, okay, let's see, and we knew that RDS is not expressed in the mouse. So can we deliver RDS, normal RDS, so normal peripherin, into the mouse retina, and would it rescue the phenotype? So that was our starting question, and And so this is the rod outer segment, and within the rod outer segment, peripherin keeps the discs tightly sealed together, stitched together, because within the outer segment there are almost 1,000 discs, 1,000, and they have to be tightly packed. And peripherin and ROM1, these are the two proteins which does that job, keeps it tightly packed. And if you don't have peripherin, like in the RDS mouse, the discs float apart, and the outer segment is never formed in the RDS mouse. It doesn't have the outer segment, but it has all of this part. So let's say half the cell is okay, and the other half doesn't happen because of the mutation in peripherin. The outer segment is never formed. So you look at, so this is again just showing you, and here is the peripherin localization at the edges of the outer segment, because that's where peripherin is localized, in the edges of the outer segment. And that's the same explanation. So in the outer segment, and this is where peripherin is not working. That was a slide you couldn't see before, but now you can see more clearly the outer nuclear layer, inner segment, outer segment, and the RPE cell layer. And here is a normal mouse, the outer nuclear layer with the photoreceptor nuclei, inner segment, outer segment, and RPE. And in the RDS mouse, where the outer segment is all but gone, so here is the nuclei, they are all present, and as I said, half the cell is there, but this is not there, and that's what we want to rescue. So by putting peripherin, would we get the outer segment formed? So it was quite a challenge, first of all, and we tried everything in the beginning, trying to inject from the front and into the vitreal fluid, and then subretinally. And eventually we succeeded in subretinal delivery. So here is the RPE cell layer, and this is the photoreceptor cell layer, and here is our injection, and we succeeded in that. And here you're showing you how the eye looked. And here a few photoreceptor rod cells which have taken up the vector, which was adeno-associated virus, and stained. But the transduction efficiency was very low, which has of course now improved, but in those days it was very low. So a normal mouse, the outer segment is perfectly formed and stacked, the discs are perfectly stacked, everything properly aligned, and here is the RDS mouse, no outer segments at all, and just the cell body is present. And then after the peripherin injection, so we cloned the normal gene into AAV2, and then subretinally injected the adeno-associated virus, as I showed you in the subretinal space, and then the virus transduce photoreceptor cells. And if peripherin starts to work, then the entire genetic pathway has switched on, and the outer segments should start forming. So that was our hope. And yes, after injection, after 6 weeks after injection, we could start seeing outer segment formation in the RDS mouse. These are the mutant mice. And I'll show you higher magnification pictures. So here is a wild type outer segment, and here is a mutant. It's probably forming. This is the tip of the outer segment, which is interdigitating with the RPE cell layer. These are the microvilli of RPE, which is surrounding the tip, as it should happen in normal retina. And clearly, through gene therapy, through subretinal injection, we were able to switch on the entire genetic pathway so that the entire outer segment is now forming. And when we did, so this is again showing the same thing, This is the RP cell layer and the microvilli on both sides of the outer segment. And this is the ERG. So here is the normal ERG, and this is the RDS mouse ERG, which is flat. And after injection, first of all, remember I showed you that earlier slide, this one. From this, what I forgot to say is that only 1/3 of the globe actually is transduced. So you will never get full function like a normal ERG. You're only going to get partial ERG, and that's exactly what we were then seeing. So these are the kind of partial ERGs. So whereas without treatment is flat, there's no ERG in the RDS mouse. But when these structures do form, an outer segment does form in 1/3 of the retina, then you begin to see an ERG. So obviously gene therapy as a proof of principle obviously works, and there's the eye just showing you that. And this is cell therapy, and this is just one slide showing you that we were able to reprogram from embryonic stem cells to photoreceptors. And here is such one example of that, which looks, this is a photoreceptor from a rabbit, and it looks very similar. And we successfully transplanted these photoreceptors, these cells, into mice retina, and they integrate very nicely, and you do get functional recovery in mouse models. No human work has been done as yet, but I'm pleased to say that gene therapy in humans, in very limited sense, is working. The very first trial was undertaken in our hospital by Professor Robinali, and he did RP65 gene therapy. These are LCA patients who could then see, and gene therapy was successful. So in the mouse model, proof of principle, it worked. We published that in 2001, I think, at that point, showing that it does work in animal models, and that gave us the impetus to carry on and finally do it in humans. And I'm sure that one day cell therapy in humans will also come. But of course, for all of this work, you need very good animal models. And what do you think about this? I think it's very important, no, to have a good animal model. And over the years, these are many of my own people on this side and many of my collaborators over the years who I've had the good fortune to work with, and very, very good people over the years have worked with me, and we have been able to do what I just showed you. Thank you.